Okay. So students, today we'll be talking about the monohybrid cross. To study monohybrid cross and analysis or its validity through the chi-square test. I'll give you one example and you can solve for dihybrid once the things are clear to you. So what we have done, we have studied already, we have done Mendelian inheritance where we studied the cross between tall and dwarf plant. That is a monohybrid cross where only one trait is considered for a parent, and the tall parents, they are tall plants, they are breeding true. They breed true. Similarly, the dwarf plant, which is breeding true, that is taken as a parent. That is a recessive parent and this is a dominant parent. And the cross between the two is being represented by 60 beads, which are representing 30 tall plants in one container, and in other container we take 60 beads which represent 30 dwarf plants. Is that clear to you? Then we go for gamete formation. We're taking out single bead, red bead, in putting it in separate container. 4 containers we take for example, and that would give you the result that these are the gametes, capital T's and green, red ones representing the small t's. However, it is going to be similar, and then you put one green bead from one container and red bead from other container, and that would give you F1 generation. So we do not have to repeat this experiment. We know that it is going to be hybrid tall. Now the question is that we'll go for the F2 generation. How we make F2 generation? Now you have in a container, let's say put all the 60 red beads and 60 green beads in single container. That is representing your F1 generation. And now shake this container for a few seconds, and after this what you do, as you know, you have now 60 green beads and 60 red beads. That is actually representing the F1 generation. This is F1 generation and having these parents to begin with. Now take out 2 beads randomly without seeing and note down its color, the colors of the beads, right? And what you will get ultimately, you're going to get these kind of combination. Greens, 2 greens, you may have green and red, and you will have red and both reds, right? So this green, 2 greens, they are representing basically two T's, and this is a hybrid and this is the recessive one. So these combinations, basically you will get 3 different combinations. And suppose now we have tall plants, tall combinations. Suppose you are getting 18 such combinations, which is pure tall, homozygous tall, and hybrid talls you're getting 29, and dwarfs you're getting 13 such combinations. And this is the genotype. This is for genotype. Whereas if you look at the phenotype, you can total these two. It is 47. Phenotype means that is appearing tall, whether it is hybrid tall or it is pure tall, and this is the dwarf, 13 number. So actual results of Mendelian monohybrid cross, we know that it is 1:2:1. So now we have to apply chi-square test. So how would you apply chi-square test? For that matter, we assume H0 is for null hypothesis. That we assume that whatever Mendel's results were, we accept that. The ratio should be either 3:1, that's for phenotype, or it should be 1:2:1, right? And there is another, whenever we perform any experiment in science, we take these null hypothesis, means we accept there is no deviation, right? And the second one is altered hypothesis, means there is deviation from the actual results. So to verify this Mendelian monohybrid cross, we would apply chi-square test, fitness test we call it as. So for phenotype, I am showing you, this is the tall plant, this is the dwarf plants. We have, as you saw, these were 47. Here it was 47 and 13. So I'm writing it 47 and 13. Expected value, what would have been expected value? It should have been actually 3:1. So if we have 60 plants, it would be 45 and 15, right? That makes it 60. Next is you write it observed value as O you write expected value as E. O minus E is, you will calculate 47 minus 42, 45, it is 2. And the other one is 13 minus 15, it is minus 2. However, you can ignore this because ultimately you have to calculate square of it. 2 into 2 is 4, right? And minus 2 into minus 2 is 4. Now, the next, what we do, just show you on the next page. This is I have calculated, that is your O minus E, right? Value of 4 by, where I was showing you, value of 4 by 45 and 4 by 15. That comes out to be, this is, you can calculate for yourself, 0.088 and 0.266. So your chi-square is sigma of O minus E square divided by E. That comes out to be 0.354, right? So now another thing we need to know here is degree of freedom. What is degree of freedom? As you saw, there are two rows. That is, there are two variables. That is, therefore, it is 2. Degree of freedom would be n minus 1. So n is 2 minus 1. So degree of freedom would be 1. It is equal to 1. Now what is the use of this degree of freedom? Now this you have to look into chi-square table, where you would look at, I'll just show you, or I'll share one screenshot with you, that chi-square value that comes out if it is higher for 0.05, right? If p-value is higher, means this value is the p-value. If it is higher than that of the what is given in the table, you have to check the table in degree of freedom under this 0.05. It means 95%. In science, we take anything is significant which is 95% significant under this particular value. So if this is higher, then we would reject the null hypothesis. But because this value is lesser, so it is not acceptable, right? So that is about this phenotype. Similarly, you can calculate for genotype. Genotypes I have written. These are dominant, homozygous, dominant. This is hybrid tall. This is the dwarf. So suppose observed value, I already show you. Here I had shown you. This is the observed value. 18, 29, 13. Similarly, expected value would be, because the ratio is 1:2:1, it is 15, 30, 15. So you can calculate 3, 1, 2 and 9. Square of 3, 9, 1 ka 1, 2 ka 4. And then you can calculate these values which come out with this. And chi-square you would calculate, which comes out again 0.889. Now, how you would calculate degree of freedom? You have here 3 rows. That is, you have n minus 1 would be, n is 3 minus 1, that is equal to 2. And you have to look for this value under degree of freedom 2 for 0.05, right? So this p-value at 0.05 reflects 5.911, which is higher. So this is again, null hypothesis would be acceptable, right? This is how you calculate it.